Okay, well, let's begin talking about soil biology by first thinking about the soil itself. And here is a kind of a broad view of soil, and I think of an important characteristic of soil as a habitat for organisms, that it has structure. And that's maybe to compare it to something like lake water or river water, which is basically unstructured and things can move about freely. And that's important for a number of reasons that we'll look at. And a couple of things that kind of relate to that, some quotes from some famous people, Aristotle back around 300 BC, and then a few years later, Douglas Adams, the science fiction writer, who many of you may be familiar with. And each of these quotes really does have an important relationship to soil. The whole is greater than the sum of its parts. And Douglas Adams, his thought was that if you try and take a cat apart to see how it works, the first thing you have in your hands is a non-working cat. So both of these relate to soil because the soil does have this characteristic of structure, and we'll see what comprises structure in a little bit. And once you take that structure away, you really remove what makes soil soil, and especially how it controls organisms and how organisms interact with soil. So this, especially this quote from Douglas Adams about taking the cat apart to see how it works, is going to be something we'll think about as we go through the course, and really what we know about soil, because this is really what we do. We take soil apart to study it, but once we do that, we've really kind of done this. We bring ourselves to the stage of a non-working cat. So how does soil structure affect the biological activity? This is probably a good place to start thinking about it, all the different organisms that are potentially in soil. And we'll look at most of these. We probably won't get into most of these bigger critters here. So these guys don't actually live in soil, but they're feeding off of other organisms that do associate with soil. And what the main thing to take from this is that we have this range of organisms that goes up this food chain, and we'll talk about food chains a little bit later. And all of these are interacting somehow. So these just show sort of the organisms between interactions between organisms. So down here at this lower level, we have this stuff in soil, the organic matter, serving as food for the fungi and for the bacteria. Actually, the plants themselves can be serving as food for fungi and nematodes and also for bacteria. They don't show that here. But then these guys down here end up serving as food for all the things up here in some way. So here's this food chain idea. But it shows interactions between organisms. Let me kind of clear some of this stuff off. See if I can do that. Yep, there we go. And so we're looking at interactions between organisms here across this whole thing, but then we also have this interaction of organisms with the soil, or with the plants. Okay, going that direction. So there are these range of biological interactions and physical interactions, and structure is going to be important in controlling these things. And one of the things that isn't really apparent from this, and one of the reasons, is that these organisms vary very widely in size. So this is a log scale. So here's one. This is in millimeters. So 1 millimeter going up to 10 millimeters. So if you were to go, maybe if I were to draw this a scale, 1 millimeter might be something like that. 10 is something like this, and then 100, probably longer than I can draw it here. So this log scale, let me get rid of that stuff, is this puts everything on a range that you can look at everything at in one view. Otherwise, you really couldn't see most of this stuff. So we have this range from things that are, this is 1 millimeter here, down to 0.001 millimeters, or this is also 1 micrometer. And down at this range we have bacteria, and then these things are going up. This is the category of microflora, and then microfauna, and then mesofauna within this range of from 0.1 to say 10 millimeters, and then the larger organisms here. So just the fact that these organisms vary in size is going to have important implications about how they interact with each other and interact with soil. And why is that? So here's an example of soil. This is probably something that's on the order of a millimeter scale. So this would be maybe 1 millimeter up and down here. So a millimeter squared. Excuse my bad writing here. I'm going to try and write a little bit more clearly. Let me turn that off here. So I'll make this 1 millimeter squared. 1 millimeter cubed, actually, since it's a volume. This is a computer simulation, so this is really just the computer's idea of how this might look. But if this is a millimeter up here, you can guess that these pores like here may be, a fraction of a millimeter, so that'd be 0.01, who knows, small. And these smaller ones here, 0.01. So these pores are going to be where organisms are living, right? So they're, for the most part, not going to be living within, well, they could be buried within the solid material, but the pore space is really going to be important because that's where a lot of the action happens, things are moving around. And if you're one of the larger organisms, for example, maybe one of these nematodes, you might be something on the order of something like this. Here's your nematode. Yeah, here we are. That just trying to move through small pores like that would be impossible. Right? So the nematode really doesn't have a way to do that. If you're an earthworm, even bigger, let's draw the earthworm over here, probably off of the scale, something like that. The earthworm really kind of plows through this and will change the structure itself. It's kind of by the way it moves through. So you would need to have larger pores for the nematodes to move around in. Why my pen isn't working here? Okay. There we go. So larger pores for the nematodes to move around in. The bacteria could be anywhere in here for the most part, except we'll see that there are pore scales or pore sizes within soil that are probably even too small to allow microbes or allow bacteria to reside in. So structure is important in controlling the distribution of the pore space, the porosity, and because of the variation in the porosity of the pore space, you're going to have variation in the distribution of organisms. Okay, so the next thing to think about is how the pore sizes may affect smaller organisms. And in this diagram, what we're looking at is a scale that's kind of zoomed in from 1 millimeter down to about 1 nanometer. And this encompasses, on the organism side, some smaller eukaryotes. So this would be small protozoa, for example, and perhaps nematodes. And in the prokaryotes, we have both the bacteria and the archaea as two forms of prokaryotes in soil. And then below that, the viruses, and then other smaller materials, proteins, and then soil, wide variety of proteins, but enzymes are going to be an important group we're going to talk about in this class, and then small molecules and atoms. So the key thing to take from this is that if you look at the other side of the scale, here are the sort of the soil terms of the pores of different sizes: micropores smaller than 2 nanometers, mesopores from 2 to 50 nanometers, things that are above 50 nanometers as macropores. That the pore scale here really is continuous, right? So the organisms kind of break at certain places, but porosity is a continuous scale. So these are a little bit arbitrary. But the main thing to take from this is that the pores that are smaller than about this size are going to effectively exclude all of these larger organisms, and especially this group, the prokaryotes, the bacteria that we'll think mostly about, as well as the archaea. So there's potentially a very large area, or some space at least, in soil that is basically not a habitat for microbes. So what is that going to mean? Okay, let's take an actual look at some soils here. Okay, so in this example, what we have are sort of three model soils. So something called clay, sand, and a loam, and we'll talk in a few minutes about how we come up with these terms. But for now, the thing to think about is just how these three different soils compare in this characteristic of the total amount of porosity. So this would be the fraction of the of the volume of a soil that is pore space. So in this case, 60% would be pores, and the other 40% is solid material. So you can see that with clays, you have the highest amount of pore space, and that goes down to sand at the lowest and loam in the middle here. And we'll see later on why that is, why these three different soils. This is called the texture of the soil affects the total amount of pore space. But if we look now down here at this graph, what we have is this total amount of pore space. So, for example, 60% in the case of the clay here. 60% of this pore space is now divided up over different pore sizes, different pore classes. So we have down here at the very smallest end, so pores of 0.2 microns, so the scale is in microns, 0.2 microns or 200 nanometers. About 30% or a third of the total amount of all that pore space, 30% of this 60%, is in this small fraction. And if you compare that to the loam, which is about 20%, and then in the case of the sand, it's something like, maybe a little less than 10, maybe 8 or 9%. But remember, if we go back to the to that prior slide, that anything in this size category of something like 0.2 microns or smaller is really going to exclude anything smaller or anything larger than bacteria. So this becomes a size of pores in this range that really is pretty much devoid of organisms, right? If we think about microbes as really kind of the smallest organisms, excluding viruses, then there's a potentially a fairly significant amount of this pore space in soil that doesn't have organisms present. What we're looking at in this slide now is those three soils, the sand, the loam, and the clay, and their total porosity expressed in terms of surface area. And what we're comparing, or what the investigators are comparing, is how much of the total surface area is occupied by bacteria. So that's what they're measuring here. So they were able to go in and count the bacteria and assume some sort of model size for the bacteria, come up with an area calculation for each bacteria, multiply it times the total number of bacteria, get a total bacterial area, and then divide that by the amount of total area that they could measure or calculate in soil, and came up with a percent of how much of the total area is occupied by bacteria. And what's instructive here is the amount of this coverage is almost always, or is always in this case, a fraction of a percent. And this is one study which has come up with this sort of value, and there are others that have found similar things. But it tells you then that that the soil is not uniformly covered with bacteria. There's actually a fairly small fraction of the total area in soil in which microbes are living. And so you think about the pore space in soil as a habitat that really it's not being uniformly covered with organisms. That there's some factors, the size of the pores is one of these, and there are others that we'll talk about later on that are controlling the distribution of organisms. So there's not a uniform coverage of bacteria in soil. So here are some images of some actual microscopy of soil, sort of like what would have been done in that last study we looked at. These are actually some different investigators down here, Noonan et al. But what they had done in this study is they took a core of soil, and here's our core of soil, and they had cut slices through the core and then taken the slices out, and they actually would have been round slices, I guess, if this is a core, and stained these slices with materials that would make cells, and that's what we're looking at here. Some cells show up as a bright blue under fluorescence light, and then they examined the slices and tried to determine what the spatial distribution was of bacteria in soil. So here's a broad view of the of one of these slices. So the scale here is 100 microns, and so bacteria. This diagram here, or this insert, is this bit here. So bacteria around maybe 1 or 2 microns. So they kind of show up as tiny little dots in this. But the main thing from this is that when they were looking at these sections of soil, that again they weren't seeing this total coverage of this area by bacteria. They could find little spots. So here's where this spot is zoomed in, where they could find, little patches. I'm not even sure if this is a bacteria or not, but here's bacteria here, and here's some here, and maybe some of these other things are bacteria. You get kind of an idea of when you're looking at soil with these sorts of stains, that it's not always totally clear what you're looking at as an organism or not. You get a lot of this kind of background glow. But in any case, this kind of distribution really is consistent with the kind of calculation that we were just looking at in the last slide, that you don't have this entire area of soil that's being kind of uniformly covered by organisms, bacteria in this case, more or less like a spotty sort of distribution. So that's important because there's really kind of a concept out there in the literature about distributions of bacteria, how bacteria grow, and you'll probably see this term, or maybe you have already seen it somewhere in your readings of biofilms. Then this is an example of a biofilm. So here are bacterial cells in this biofilm. And then this other material is kind of a gooey substance, gum that the bacteria release and allow them to stick. But biofilms are important in a number of environments. A lot of environments inside of us, inside of our intestines, on our teeth, contact lenses, and forming on rocks and things like that, in rivers. But this idea of a biofilm probably isn't very applicable to soil, because you just don't seem to have this kind of a wide coverage of bacteria in soil particles. Let's return and look at the physical structure again of soils, and here's our model. It shows the porosity and the pore space in soil, and this is the thing we've just been talking about that has some effect on the distribution of microbes in soil, and larger soil organisms as well. So what are the physical aspects of soil that contribute to the creation of this structure? So to begin with, let's just think about what soil is. Take all that stuff off, and in a very broad model, which you can see if we divide up this is our model soil volume. Go back in, and let's try this one more time. There we go. And here's our model soil volume, and you can divide up this piece of pie into kind of roughly 2 halves. One half is the solid phase, and the other half is the open phase, or this is the pore space we've been talking about. And in the solid phase, it's going to be divided up between mineral material that's derived from the original rocks, and then the organic material, which is going to be coming from primarily biological sources, not primarily, totally from biological sources, but primarily plants. And plants are going to be the actual origin of all this organic matter. These amounts here are variable, so this is really kind of a rough breakdown. These things are all going to change with different kinds of soils and conditions, but just as a sort of a general model. So we'll talk first about this mineral part here, and how this mineral part has some effects on the on soil. So here's the way you generally would look at the mineral phase of soil. These are called the soil separates. These things here, the clay, the silt, and the sand, or they're also called the particles. And these are just a size class, right? So here's the particle diameter up here at the top. So anything smaller than 0.002 millimeters, so this is 2 micrometers, 2 micrometers, is called clay. Anything in this space between, or the size fraction between 2 microns and 50 microns, 50 micrometers. And my writing here is not so great. 50 micrometers is called silt. And anything larger from that, 50 micrometers to 2 millimeters, is sand. And of course, there's things that are larger as well. This gets to gravel, so forth. But again, these are just size fractions, right? So you can say take any of these particles here and grind them up a bit and get them into this size fraction, and grind them further and get them into that size fraction. Wouldn't have changed anything about what's actually making up this piece of rock, right? It just becomes a smaller size fraction. Okay, so that's important to remember. So one of the ways that the soil particle information is useful is to derive the soil texture identity, and that's done by using this textural triangle. And this is probably one of the first things you would learn if you took a course in soil sciences, soil textural information. And the way this works is, if you know the percent silt, you always sort of start with this going from right to left. If you know the percent silt at some point, 30% for example, you would bring that in. And for example, if you had another 30% of sand, you would bring this in, and right around here where your balance of 40% is, put you right about in there for a clay loam. So that's how those previous people had come up with the names of their soils, as clay, sand, and loam were just the relative amounts of these soil particles. So this is important because you'll see these names pretty often. Clay loam, for example, is something that you may see as in any papers that you might look at during the course of the semester, and that's where these names come from. It's from this textural triangle. But the other aspect that's important as far as the structure goes and the porosity is that the size of these different particles is going to affect how they can pack within a given volume. So you can see that for example, the sand as the the sand as the biggest one here. This is the circle is going to be our same amount of volume for all of these. That the large sand particles, just a fair amount of the space is taken up just by the solid mass of the sand particles. If you were to then break this particle up and grind it up into, some amount of silt, that space which was previously occupied by this sand particle now becomes some amount of pore space, right? And if you were to do the same thing with all these little silt particles and grind them up further, and you would make clay particles. And so what you've done is effectively transform solid mass, where there's no space, into space, right? Because you've broken up the particle into smaller and smaller particles. So how does this work? Which way do you think porosity increases? Increase in this direction or increase in that direction? If you voted this direction, you're right, because the higher the amount of clay-sized particles, the greater amount of pore space you'll have, just because of this aspect of the clay particles being smaller than anything above them, and thus you'll have more pore space potentially, right? So all that solid space that was occupied here now becomes open, right, within these clay particles. So the other part of the soil that is important, go back up again, is in terms of the solid phase, remember we're just talking about the solid phase now, is this organic matter fraction. All right, so you all know organic matter. If you ever have taken a piece of soil up on your hand, you would see that it has some of this dark color, either this or maybe a little variation in the color of the soil. If you were to dig a pit, you would see this. This is all the organic material sort of accumulated in the surface, and this decreases as you go with depth. But the organic matter is important. As a fraction of the whole soil, it's fairly small, but it's one of the key constituents of soil as far as life is concerned. And we'll look at some reasons why that is. Think about back to our earlier slide. We had this diagram, and down here, this is the organic matter. Oops, I'll get this right. This is one of my earliest screencasts, so you're going to have to bear with me a little bit here. But here's the organic matter that we're talking about. So this is that stuff that's the brown material we were looking at in the previous slides. And this is serving as the base of food for all these things that are down here, which then serve as the base for food for all these guys up here. So it's critical as far as the life goes of the soil, okay, just in terms of this food chain aspect is concerned. You think about how soil organic matter is distributed. So here's a map of the United States, and this shows organic matter distributions across the country. And so very high out here is the darkest parts, and low in the gray. So you can see kind of more in our desert areas, there's not much. But right in here, not too far from where we are, is one of the highest levels of organic matter in soils in the country, with the highest concentrations of soils with high organic matter. And so these are the very fertile soils out sort of in the western part of our state and in Iowa, which are really among the most fertile soils in the world. So that high organic matter content is important for plant life. So the plants help produce it, but they also rely on this organic matter for food. All right. So, and you can kind of see how this varies. All the agricultural areas across the country, where they're most productive, that's where you'll find the high organic matter, soil organic matter levels. So in our earlier slide, looking at the distribution of microbes in soil, we saw that they were pretty much scattered throughout the soil pore space. There wasn't a uniform distribution of those. And the same appears to be true for organic matter in soil. So although the soil may look fairly uniform in terms of a dark color that you may see, or some color of brown, it's probably not a uniform coating of organic matter everywhere. And so this is some more current analysis of soil. And this uses a technique where the soil particles can be visualized directly by something where the elements can be imaged. So oxygen is turning up as blue, and so oxygen is silicon oxides, or what the soil particles are composed mainly of. So that's why blue is predominant through here. Carbon then is going to be representing the organic matter, and we don't need to worry about all these different numbers, but just kind of take it that anything that's yellow here, or green, or even the pink is sort of tracking the carbon. Let's just look at one of these for example. So here, I forgot what the scale is here, but this is something on the order of maybe microns or so. But the organic matter you see is really kind of accumulating in certain spots. And you don't see these things as sort of uniformly colored yellow. So the organic matter distribution then becomes something that's important, because most of these soil organisms, the microorganisms, the bacteria in particular, are going to be relying on this carbon as their food source in soil. So while one thing that affects the distribution of microbes, especially bacteria in soil, is the size of the pores, the other thing may be the carbon locations, because that's their food source. And to some extent, these two things are going to be interacting. So the pore size is going to some degree affect the distribution of the carbon. This kind of gets back to our cat, remember? So we talked about the cat, that once you take it apart, you really can't understand how it's working. Here's the cat taken apart. So these things were all put together originally in the soil, and somehow in their original composition, this led to this distribution of organic matter. Why? Why is it like this? Well, we can't really say from this kind of observation. So this is our non-working cat in a sense. We can observe this, but we don't know why this has occurred. We can develop theories about what's going on, but we don't know. But this is one of the first examples of how we can take things apart and not really take soil apart, then learn something about it, but then really not fully understand how things are working in nature. So let's return to the food web for just a minute. So what we're just talking about then is the organic matter, this material down here. And that this then is critical for the support of life, both because it supports the plant life, which is also the source of this, but also because it supports all these guys that then support the life of these things, and which also then have important effects back to the plant. So this organic matter in the soil becomes a very much of a central component to soil functioning and to life, because we're out here somewhere, we're living either directly off of those plants, or we're having animals that live off of these things, and then we eat the animals. But in any case, we are as dependent on this stuff as anything is out here. We just don't maybe have so much of a direct contact with it. It's probably a little bit more of a hotter topic than you might expect. This kind of gets us into thinking about the Martian. I'm going to take this off. Back to this. The Martian. You know this guy. This is the guy Marvin the Martian from the old Disney cartoons. So what does the Martian have to do with soil organic matter? Well, if you saw the movie, it's not that Martian, it's this Martian. And if you saw the movie or read the book, best movie on this or any other planet. How about that? Here's Mars. All right. So how does Mars look to you as far as some place to grow crops? Well, hard to tell from this, but the one thing you don't have in Martian soil, if you want to call it that, the regolith, I guess, is organic matter. I mean, there's nothing out here that has produced organic matter that is going to then create organic matter in soil. So this is all purely mineral. And so one of the key things about supporting life on Mars, and this is what happened with the Martian character in the movie, is they had to do something to transform the soil itself to grow crops. So if you remember what our Martian character did here, this is our Martian here. I've forgotten his name, but he was sort of farming inside of his little tent, and what he ended up doing, if you remember the story, he was recycling his own waste into the soil to grow potatoes. So he was living off of the potatoes. And look at that. Well, where did that come from? Does that look familiar? Yeah, that's organic matter. So somehow he managed to transform the Martian soil into some kind of organic matter material. But it's really central. I think if a movie like The Martian can pick up on this idea that you need to put organic matter into the soil to support plant life, then I think you really kind of get a sense that this is really more in our culture than you might realize. So we'll be talking more about soil organic matter in the future. And I guess the question is, you maybe think about is, so why is this so important? What are the things that soil organic matter does, or what makes it up, that make it so central to plants and to us and Martians?